PhD from Banaras Hindu University, and afterwards she has postdoc fellowship at CIML France, followed by Colorado State University in US, and then National Institute of Health, NIH, USA. And later on she had been professor at BITS Pilani, and then after that she is at CBLU. So she is working on the mosquito immune system and interaction with the Plasmodium as well as dengue virus. So I would like to invite Professor Lalita.
Thank you, Professor Singh and Professor Sanjeev Kumar. It is my great pleasure to be present over there. I would like to thank the organizer, Dr. Alok Bharti, for giving me this opportunity to present my research work. Here I am switching from COVID-19, which is an excellent work done by Dr. Bhatt, who was here, to mosquito immunity. So it's like a kind of switch, and I hope you are able to understand that though we recently gone through this COVID infection, mosquitoes and the diseases transmitted by mosquitoes are also very, very important. During that time, I remember, like when COVID is going on, so many dengue infection is going on. It's very difficult to find it out. It's really a dengue infection or it's like a COVID infection. Everybody was like kind of scared. So today, what I'm going to present over here is about the transcriptome analysis of ABC transporters in immunity against blood-borne pathogens. So before I would like to give some introduction and why we are working over here, what is ABC transporter and what we know about mosquito and its immunity. So just now we have learned about like we humans are vulnerable of different diseases. Here we talk about direct infection, which is like a COVID. We just learn about how this COVID is affecting us. Here are some blood-borne pathogens where we talk about indirect. Here we require two hosts. So when we talk about two hosts, like here, in case of blood-borne pathogens, we are talking about the one host is mosquito and the other host is the human. So I'm giving you two pictures just as a glimpse. Like here is a malaria, which is transmitted by Anopheles stephensi, and there are several species of Anopheles. And now the other side is the dengue, which is transmitted by Aedes and many species of Aedes. So there is always easy to target the human host. But when we talk about the mosquito host, it is important because if you think about, like here I'm giving just examples of several other species which transmitted disease when even killing the people. Mosquitoes is one of them. And because they are able to transmit dengue, malaria, West Nile, chikungunya, and variety of bacterial and viral infection and plasmodium infection. So I would like to go why we are why we are looking for mosquitoes. Because when they transmit several diseases, there are different ways to control it. One way is control is just for protection, as we all know that we can get the vaccinations. On the other ways, like we have got different anti-malarial drugs and other drugs which we can protect ourselves. On the same time, we just looking at the transmission blocking. So if we able to block the transmission from mosquito host to the human host, that will be easy. So we can not allow parasite to go inside the mosquito. In that case, this term we use as a transmission blocking. One way we can just even kill the mosquito through the insecticides. On the other way, we can prevent ourselves, just we are using different like bed nets we are using repellent. And one of the very important and emerging one is like a blocking parasite development inside the vector. And when we say inside the vector, we have to know more about vectors. So I'm just giving you first two glimpses of one. I worked on like a little bit on killing the mosquito because this is easy, we able to do experiment very easily. And then we move towards the blocking the parasite development. So you can think about the mosquito immunity and how they able to control parasite development inside the mosquito. So just giving a one insecticide example. In case of that, we were looking for when we came from US and with Filani, like we were just like setting off mosquito lab is not very easy. So we were setting the mosquito labs, same time the parasite and everything, and at the same time we were saying, okay, let's look for any biological way which are environmental friendly, we can control the mosquito population. So here I am giving the one example of agave augustifolia. This plant is present in the Rajasthan area and towards the desert, and very specific that when we take just water extract, they have a efficacy to kill the mosquito larva up to 100% within 48 hours. So, and important thing is that they give the activity only in time of summer. As soon the winter comes, no activity. So we were surprised that the student might not be doing correct experiments. All of a sudden it's required that we thought, okay, let's change the temperature. So when we change the temperature 37 degrees, again this plant extract is giving the results. So we published over there in the journal Frontiers, and now even government is also wants to know about this plant, and they are looking more how we can find it on different extracts and use it for just killing mosquitoes. From here I am just switching towards the mosquito immunity. So once we know how the mosquito immunity is able to kill the parasite, we can easily block the transmission. So mosquito have a very strong immunity. One is called pattern recognition receptor, so its innate immunity is present. And they also have a signaling molecules like immune signaling pathway. I am working on JAK-STAT pathway. So JAK-STAT pathway is very well characterized now in mosquito, and we found that this JAK-STAT pathway is actually working as a negative, positive regulator, is actually suppress the plasmodium development. So just for students who are not in this field, this plasmodium, they enter into the mosquito and it almost takes 21 days to stay inside the mosquito, and the mosquito is able to transmit the disease. And we are looking at the stage, because this is the midgut epithelial, and we are looking in the stage when this becomes a oocyst. So it's better to, if it is possible, we can control this transmission because it is a midgut epithelial, and if we control over here, that is much easier. So I'm just giving an example of STAT signaling pathway. We silence the STAT molecules, and when we silence the STAT molecule, we find it out in case of Ghorai, we find that this is increasing the parasite load. That means we calculate on each individual mosquitoes the number of oocysts present over here. On the other side, we find it out that in day 7, when we talk about the day 7, it's like of SOX gene, which is the suppressor of STAT pathway. This is going down. That means it's suggested that STAT pathway is activated, and when we silence, that the parasite number is going up, and when we have a SOX, then the number is going down. And this interesting finding, we found out that not only the early stage of oocyst, but even the late stage of oocyst, this STAT signaling pathway is there. So from here we are started looking all more about the STATs, and here we find it out like there are several STAT molecules are present in 18 Anopheles mosquitoes. Some having a STAT A as a conserved one, which have a different introns, whereas a retrotransmission there is a STAT B. And actually STAT B is regulating the STAT A. But in few mosquito species like Anopheles sinensis, Dyrens, Dylangi, Albinus and Ferropi, they have a single STAT. So when we talk about any like control mechanism, and we say it's easy, now we find it out molecules and it can be easily controlled. We find out there are so many types of plasmodium present, and there are different in mosquitoes. So combination of all these to have a mosquito and parasite and finding out one target molecule, because still is going on, still we are looking for. After that, we move to one of the molecule which is present inside the mosquito, and that is also an immune molecule, that is apolipoforin, and here we also did the silencing experiment. It's a big experiment, it takes like almost 12 days, because we have to use the double-stranded RNA, inject inside the mosquito, gave the plasmodium infection and allowed the mosquito to survive, and then started counting the parasite. So I am giving here the example of apolipoforin 3. This is known for flight lipid proteins, but they are also play a very important role in mosquito. So here when we see that laxity and when there is a apolipoforin 3, the infection is going down. So that is actually working as a negative regulator. So when there is a no apolipoforin, parasite number is going to be increased. So it's actually helping parasite to grow. So here I'm just saying that we are working hard to find out few molecules which going to target towards the plasmodium development inside the mosquito bite. And in that case, we are finding new and new molecules. Now I'm coming from here towards the ABC transporter, which has been recently started working, and these ABC transporter, why started working? Because we find out some ATP transporters, they are actually having a role in insect immunity. Some ABC transporter immunity, these are the some papers which coming out. And from here we also search the literature, and this we find out that some like in case of wheat or in case of other molecules, they are actually transporters are sensitive to or suppress the pathogen development. And single mutation in wombat we find out that with the ABC transporter resistance to the BT toxin. And there are a few more examples where we say yes, these ABC transporters are working. I'm just giving, I think enough 2 minutes. Okay. So here we would like to know how this ABC transporter, which is ATP binding cassette transporter, and whether it's working in mosquito immunity. So this project was funded by DBT, and we are looking for some novel immune molecules which may regulate the pathogen development and might offer a new insight to interpret the mosquito cycle of bloodborne pathogens. I'm giving this example how this ABC transporter looks like, and in Aedes aegypti. So ABC transporter have been 2 domain. One is called nucleotide binding domain, and other one is transmembrane domains. This nucleotide binding domain is actually having a ATP binding, like you can see over here, and they have a transmembrane domains. So transmembrane protein that transport amino acid, sugar, peptide, small lipid across the biological membrane. And as I said, they have a NBD which having a Walker A signature sequence, Walker B motifs, and they have a signature sequence. And TMD, they have a 6 helical embedded in the bilayer membrane. So this is a ABC transporter proteins. Based on how many ATP binding cassettes they have, there are 2. One is called half transporters, or you can say full transporters. You can see this is the half transporter and the full transporter. This is the mechanism how it working. That molecule is coming, ATP is and able to release the molecule over there, and it's released ATP. So what we are looking over here, so I am talking about Aedes aegypti mosquito, and in Aedes aegypti mosquito we are first identifying and characterizing the ABC transporters, and then we are looking for their expression in the mosquito development and identifying their expressions and find with the immune challenge. We are giving the immune challenge of bacteria over here and finding out if there is any molecules. So reverse genetic approach we are using to find out the specific molecule of ABC transporter. So with the literature search and the database present over here, we are looking for ABC transporter, and in case of you look over here in Aedes aegypti, there are almost 59 ABC transporters are present, and they are having a subfamily from A to H. We compare like in Drosophila, Anopheles, Bombax, Culex, as well as Homo sapiens. What we find out that ABC A, C, and D subfamily are widely expanded. So we published this paper last year in Pathogen, and afterwards we are also starting looking like whether this ABC transporters expressing inside the mosquito gut or in the carcass. So here are the some genes which we say, like in case of ABC, Some are expressing both in midgut as well as in carcass, whereas some are only expressed in carcass or some are only expressed in the midgut. So there are some very unique because they have a different domain structures over here. So we started targeting few of them. How we targeted? We are looking for some molecules, like here I am giving the example of ABC B2, C10, G1 and G3. We did the experiment giving the live bacteria challenge with these mosquitoes as well as heat-killed bacteria challenge. First, over here you can see the different time kinetics from 1 hour to 12 hours and looking the defensive, which is very known molecule, antibacterial molecules, and this expression is going high shifted the related mRNA expression. B2 of ABC transporter B2, this expression is there but not very high. Same way in case of C10 and D1 and G3. So this expression with this one, we can say that expressions goes up and down in some cases, and here in some cases you can say in C10 or G3 the expression is like going up and then again going up in 6 hours. So from there we are looking for different, this is a little bit very big slide because we are looking different ABC transporters and how these different families are working. So I am targeting only 1, 2, 3 over here, and this is the 4th one and 5th and 6th. When we challenge, so we say ABC transporter of subfamily A, C and G are induced after both the bacterial challenge, if it is heat kill or if it is a live. Then we looking for one important one that is called ABC G3 because they have a similar to E. coli toxin antitoxin system, and we find out that how they are similar to other and where it is found. So we use KEGG's sequence similarity database to analyze this ABC transporter G3. Now from here we plan to silence this ABC G3 genes through RNAi, analyze its effect on blood-borne pathogens. So here you will see that there is PCR amplification of ABC's G3 genes of 4 sets of midguts we are using over here, and then we clone it and make a double-stranded RNA of them, and finally you can see the double-stranded RNA of ABC's G3. And after that we did the silencing. So here is the silencing results. We make a double-stranded RNA of G3. This is the control. There is a 80 to 90% reduction in the mRNA level, and then we are looking for their bacterial load. So we find it out that when the gene is silenced, the bacterial load is increasing. In case of sugar fat, as well as when we give the bacteria supplements, also it is increasing. We suggested that ABC G3 is actually working against this mosquito, Aedes aegypti, and is working as an immune molecule. We look for another gene, that is ABC C12, and we did this experiment to find out their viral load when there is a dengue infection. And from here, this experiment is preliminary experiment. We are also again looking into more details. So based on all these experiments, we are just giving that mosquito having a strong immunity, they are able to kill the parasite inside, but we have to find it out exact molecules or the different targeted molecules over there. And when we talk about new molecule ABC transporters, we find it out there are 59 ABC transporters are present in Aedes aegypti, and they have a gene or gene segments duplication. They are widely expansion of A, C and G subfamily, reveal their importance in the mosquito biology, and suppressor of immune ABC transporter is the survival strategies of the virus. There are different scopes. We can also work again currently the antigen specificity of innate immunity, suggesting to explore the list of transporters and other immune genes in the regulation of virus development, and designing the strategies to manipulate this important molecule for blocking the pathogens development and transmission. So I would like to stop over here, and I would like to acknowledge The stat work is done by Kuldeep Gupta, Apple is by Rini. Professor Sanjeev is with us, doing all sorts of experiments and helping the students. And whenever we stuck, this is my team. Here's the Niklesh who is working on this work, and this is students that I am is working on daily. Thank you so much.
On the halfway signs, please raise your hands. Yes.
Have you tried silencing the transporter?
No. It's just like first of all, like you have to target one organ. So we target the gut, salivary gland we haven't tested.
The other thing, so initially I think the bacterial infections you mentioned, you were using a mixture of live and dead bacteria.
Yes.
Is there any specific reason?
No, actually it's not very specific reason. We just would like to say as a antigen elicitors, how they are working. So if it is a heat field, that means it's going to produce molecules. When we are using live, what there is an effect. That is the one.
Any?
So Lalita,
Yes.
good lecture, good piece of work. Just a couple of observations or questions, whatever you can say. You have done work on ABC transport,
Yes.
right? So is it possible at this juncture, can you correlate the dynamics of this ACP transporter genes with the propagation or development of this pathogen, for example in malaria, mosquitoes we were talking, or you can say their transmission, I mean can it be correlated with the development of the pathogen and its transmission, transporter genes and the transmission?
Okay, sir. In that case, like we started with the just first to align all these ABC transporters present inside the mosquitoes. And first we find out like they are in, there are so many duplications over there, and almost they have like one say ABC A, there are also several ABC A transporters. Some of them we are targeted, like we just talk about G3. We give the bacterial infection, and yes, there it's showing, when we silence is showing, yes, it is working. But further we have to do more experiment to find it out how they are controlling the antigen or any virus infection.
Okay, just give me one or two ideas.
Yes, sir.
In Singapore, there was a dengue problem.
Yes,
There is one company, Oxitec, they released transgenic insects. So I think if you can catch hold of those people, I mean you can see the transporter gene mechanism in those mosquitoes
Yes.
which have been released for that purpose.
Sure.
Basically, the concept is based on sterile insect technique.
Yes.
And for the last 3 decades I have been working on that. So my interest is, if you can correlate that, then I would come to the next question whether the radiated insects can have some sort of mechanism working over there or not. And another point is that suppose you are able to find out this innate immunity, will it be possible that you can use some efficient method to block the transmission?
Yes, sir. In case of like Plasmodium, when we talk about Plasmodium, they are looking for the molecules, and especially when we are the salivary glands. So in the salivary glands, they are finding out the sporozoites during that time and how they can able to block the transmission. There are several molecules which are working over it.
Thank you. I don't know whether you are remembering or not.
Yes, sir.
This couple got the professor's position. I was incidentally the expert in that committee. So,
Thank you, sir.
Please. Any of the STAT inhibitors can also be a little bit molecule to inhibit the virus. What sir is asking to block the...
Sir, actually, when we look for the starts in the pathway, we find out the NOS. And NOS is really this very well known, and like even Sanjeev, Professor Sanjeev is also working on virus series and NOS. And that is, we are planning to really look for that as a
plant extract you are showing that has some ovicidal property also. It can kill at the egg stage.
At this stage, we
That will
haven't
be more
checked it.
advantageous, right?
Yes, we haven't checked it. But of course, it has a mosquito stage, but not egg stage. We have never checked it. No?
Can go to the larval
Larval
stage.
stage.
If it has ovicidal property, it
Yes.
will be all the more good.
Now they have seen water extract, they may see different types of extracts, you know.
No, whatever the extract we tried,
Both. This
but
is
we
the-
only stick to the water one because preparation is easy and we are avoiding the chemicals. So water one must be least.
No, water extract is fine, but,
But he's looking
instead
for
of
egg.
larvicidal, whether it has ovicidal property, means it can hatching up egg can be avoided, no?
Yes,
He
he
wants
wants.
like it.
We
We have to
can
try that.
try that.
That is more advantageous, more preferred one.
I think there are a couple of things very interesting one can do. I can give one idea at this moment.
Sure.
I may not forget that. There are certain photosensitizers and they are very well documented, but very less work has been done in that case. So photosensitizers are very safe. At the same time, India has got no dearth of sun. The moment you use those chemicals and there is a sunlight, they will immediately, they will be toxic. Alpha T has been, it has been proven a very good insecticide against these mosquitoes as well as lepidopteran insects. And we have been giving the data about bombics, and these are only two groups, lepidopteran and different. Photosensitizers का थोड़ा सा देखिए, उसको इसके साथ जोड़ेंगे, it will be great.
Thank you. Thank you.
Thank you, Professor.
I request Professor R.K. Seth to please felicitate Professor Lalita Gupta for delivering such an exciting.
Okay, we have another candidate from our department, very brilliant candidate, Dr. Soma M. Ghorai. She is associate professor in Hindu College, and the area of interest is comparative immunology, microbiology, and biochemistry. She has been a wonderful student, and she got this, research associateship from DBT, CSIR, and a very sincere student.
I would like to, I'm blessed to be here to be chaired by Professor R.K. He has been my teacher, and it feels great to come back to the department once again. And again, I would like to give my utmost gratitude to Professor Alok. When he just popped up this question to, have me on to speak something, I said, this is not exactly my field, I work on reptilian immunology. But then, yes, the COVID times has taught us many things, isn't it? So those 2 years of the college where we had nothing to do, but we had certain, the warm inside our head, and we wanted to do something, not to sit idle, and do something on nutraceuticals. I collaborated with a few of my colleagues from chemistry department, and they were synthesizing some novel compounds. And those are natural compounds which are usually found as herbs and spices, the main compounds. And those being chemistry people, they were actually looking into for the green synthesis. The way it's like green synthesis means like the way you synthesize certain compounds with the minimal use of any chemicals, so that can be easily metabolized in the environment. They are safe to consume, and they have drug-like capacity. So thanks to Professor, she's Dr. Neha Kapoor from my chemistry department, and we both worked on few things. And as I said, when the topic of this conference was told to us, it was nutraceuticals in chronic diseases. And COVID is one thing that we were actually going through this, and we just thought of working on certain nutraceuticals and how they would work in COVID. So we all know, yes, nutraceuticals, and it has been told for the past 1 day that we have been listening to so many people talking about cancers being taken care of and other diseases like leprosy. I just heard Professor G.P. Talwar in the morning these are the chronic diseases that are being taken care of, cured by the nutraceuticals. And yes, here we are talking about viruses because even these nutraceuticals have certain role in virus curbing of the virus. And here we have certain basic spices that we usually use in daily lives like cinnamon, turmeric, black pepper, basil, giloy, neem, garlic. It's everyday use, and they also have antiviral activity. So looking into that particular thing, it's purely in silico work. As I just said, it is a purely in silico work where we looked into for few important components like the cucurbitacins, quercetin, berberine, maltodextrin, curcumin, piperin and orientin. These are few compounds which have been, there are many papers by the time, quite a few papers have already been published stating their importance in having certain antiviral activity towards COVID-19 or SARS-CoV, I would say. And we listed about 32 compounds from the literature, and these compounds had certain drug-like molecules, and they can actually have they can show the effects on 4 or 5 main targets that are there for the COVID infection that can be caused by COVID infections. One is the proteases, that is the empro, and these are the replications of the polymerases, the helicases, and yes, of course, the non-structural and the structural proteins that makes the entry into the cell, the virus which makes the entry into the cell, as well as we all know, the cytokine storm that is released after the infection, and how to curb the cytokine storm. And these cytokine storms are basically taken care by the JAK-STAT pathway. So basically all these molecules can be targeted at any of the levels. So this is a common diagram that also is made by one of our, this is an amazing thing, the kids these days are very, very fast. So, and this is one of our graduate student who prepared this graph, and she made this and it was published now. And this work we actually did with cucurbitacins. And I'm not presenting that work here, it's the work we did with cucurbitacins. And we could show that cucurbitacin H, G and G, that is a glycoside form of the cucurbitacins that is found in the cucurbitacea family, would actually affect by the in silico method in all these 4-5 steps that have been shown. It could affect the entry point here, it can affect the proteases, it can affect the polymerases and as well as the helicases, as well as can affect the JAK-STAT pathway. So we then went further and looked into for all the 32 compounds through the literature, and these are the 32 compounds, and all of them we went. We had this Schrodinger software with us, and the Mastro the software where we did the docking for each one of them. And here you can see it is done for the polymerases, and the best docking score was seen by these 5 important components: arctin, verilactone, butane, amidine and curcumin. Apart from the other ones, they didn't have that better score. And we went on further, we did again the dockings with all the 32 compounds with this is done by the human ACE receptors, the receptors which is we all know where the entry happens. The spike proteins basically bind to this and the entry happens. Here also, again, we got the repeated answer of having the arctin, verilactone, butane, amidine and curcumin showing the best score. We went further, then we did again with all these 32 compounds for the the polymerases as well as the JAK-STAT pathway. Here we could get butene as one component being a common among the others, whereas for the JAK-STAT, yes, of course, we have the amidine, the verilactone, curcumin as well as butene showing. So here, in all the 4 cases, butene is one common component which has been repeatedly showing itself, and it has been able to show a good, beautiful D-score value with all of the PDB files that we could do, and did the scoring, the docking studies. Further, we took this molecule and did the further activity. We went on for checking the ADMET. Now this is ADMET is done to check the drug-like capability of any molecule. So this is again software where we see the ADMET stands for absorption, distribution, metabolism, and the E stands for the toxicity and all. So these components are looked into, there are many components, out of which it's a rule of Lipinski 5 rule which has been followed. What is that? Lipinski means where there are 5 important characters which every drug should, they should be able to follow that. If they don't follow those important rules, they do not fall under the drug-like capability. One is that it should have less molecular weight, less than 500. They should have hydrogen bonds, rotatable hydrogen bonds, acceptor hydrogen bonds, and there are certain, this is the lipophilicity and the distribution, this is the log S value. Log S value shows the flexibility of the molecule to dock. So basically these are the important 5 Lipinski rule, which these are the basic steps that we go in before we actually take any molecule to in vitro or in vivo studies. So these are the basic steps, like looking in for the docking studies and then going in for the ADMET studies. So here also we could see that acting failed in 2 main important components. It failed in the molecular weight as well as it failed in the hydrogen bond. If you have too much of hydrogen bonds, that also makes it a non-good drug, because that loses its flexibility. The flexibility has to be in a manner that they can actually undergo certain conformational changes, because whenever any receptor binds to a ligand, it needs to undergo certain conformational change, only then they can go on and activate the or do the signaling pathway. So the flexibility of the hydrogen bonds is also very, very important. Apart from the hydrogen bonds, there are other hydrophobic bonds, van der Waals forces, as well as the non-ionic forces, which are also taken into consideration. So this we did detailed molecular MD simulation activity, where this first we did with the HC, the human HC molecule. Here you could actually see this is made up of basically, there are 2 main important subdomains. This is subdomain A or 1 you could say, and subdomain B, which is separated by, but you can see the yellow helix. This comprises about 17 different amino acids, and this is this we basically did with butane, but this applies to all the small molecules that can easily dock to this particular HC molecules, which shows that this area is where basically any molecule which has molecular weight, comparable molecular weight, can dock to this particular region. And this basically shows that light blue meshwork shows that this is where the binding of any ligand takes place to the HCE, that is the human ACE2 receptor. We went further, carried on. When I go to this region, this is the part which shows quite a good docking, and this one. So little more seconds. This was done by MD simulation. Now what is MD simulation? MD simulation is something which looks into for protein-protein interactions, and where we looking for a more closer look into how the interactions are happening and at which bonds it is forming. So when we go into deeper, this is the structure of butane. Basically, this structure is a simple structure, and it can be easily characterized, easily taken, and butane can be easily even synthesized in the lab. For that matter, it is almost found in Butia. Butia is Palash flower. I think that is also known as forest of fire. During the monsoon seasons, Delhi roads are full with monsoon, that big Palash flowers, beautiful red color, yellow color, mixed orange colored flowers. And Butia is also seen in some other, butane is also found in most of the yellow and orange color flowers. So even you can find it in gladiolas, and even in the sunflower, the other, I would say chrysanthemum, the orange color chrysanthemum also has that butane molecule. So here when we see the interaction, we could see that the lysine, the asparagine, the tyrosine, and proline and aspartic acid. So basically the hydrogen bonds is seen with the asparagine, tyrosine, aspartic acid and proline. And this non-ionic bond which was seen with the, this is basically, I would say, this is the non-ionic bonds, or the butane which is not an hydrogen bond basically. So this there are many electrostatic bonds also which is important during the binding of any ligands to the this thing. So these 4 important moieties are, we can see where the any molecule, it is butane is something that we have taken into consideration, but these are the basic moieties, the amino acid moieties which actually bind to the ACE2 receptors. Further, we did MD simulation. Now this simulation is done, this is done by the simple software which is known as the, it's known as the web server, exactly the word, it's known as the Haddock web server, or where the entire study is done in nanoseconds. So it starts from the 0 nanoseconds to 500 is the maximum limit that is taken care of, where we actually see the stability of both the ligands. Now this designates butane and this designates the HC. Here till 200 nanoseconds you can see the stable condition, and later on from 200 to 500 you can see certain peaks that is happening here. So here certain flexible or movement is seen here. Now this is basically considered by 2 important methods. One is known as root mean square deviation and the other one is known as root mean square fluctuation. Deviation and fluctuations means how they are changing the conformation. Deviation, if it is less deviated, that shows that stability, and fluctuation is also very important. It's not always the stability that matters. Fluctuation is also important because that helps in changing the conformation of that area. This is very important because allosterically binding of any molecule is important because that changes it, the conformation, and helps like if we are using a small molecule and acting as and looking at as a drug-like property to bind to the ACE2 receptor so that it doesn't go and bind to the spike protein, it is important that they allosterically bind to that area, change the conformation, and not let the spike proteins to bind on it. So it's also important to have the fluctuation thing. So later on, when when we did the fluctuation, you can see certain peaks happening. You can see certain peaks, and with further investigation later on, there are other softwares known as Haddock. This is HADOCK Haddock software, which takes care of how to look in for these fluctuations and how actually they are doing the changes in the conformation change. We did this thing and we could see that at 0 nanosecond, the blue color, the cyan color, has the docking is like this, binding is like this, and after 500 nanoseconds, it has come to this yellow color docking. And you can see the conformation also has changed here. And now there are only 2 important areas where it is binding properly, this and this. Rest other has undergone certain conformational changes. So this was further taken care of. We did a little more studies, and here we looked into how basically the original SARS-CoV-2 spike protein would have bound to the the human ACE2 receptors. We see that they bind basically at 4 main important domains, and these domains are known as hotspots. You can see that hotspot A, hotspot B, hotspot C, and hotspot D. So these are a normal spike protein of the coronavirus will bound to the ACE2 receptors definitely at these 4 hotspots. Now, if we add butane, or here we are using butane or any other small molecule, the binding of the butane makes certain conformational changes wherein you can see only B and C still bound to them, whereas it has undergone a lot much of fluctuation as both the hotspot A and D. That means butane has bound more strongly to hotspot A and hotspot B. And that's what these fluctuations all show. These fluctuations, as I said, these all peaks. These are important because these fluctuations has led to the conformational changes at these 2 important hotspots and made the binding of the butane more strongly to the ACE receptors, not letting the spike to bind to it. So this is how it shows it. And further we could gather, we went a little more ahead with the MD simulations, and we could see that these hotspots bound to be mainly at the tyrosine, at the then it is this is the lysine, this is the isoleucine, and then there's a threonine, and then there's this aspartic acid. You can see these H. This blue color shows the hydrogen bonds, and these ionic bonds are almost nil. So you can actually imagine, as I was talking about that K144, that ionic bond that was already there, in between, that has been totally taken care of. There is no ionic bond. It's basically all the hydrogen bonds and the hydrophobic bonds which has made the... now, when we talk about the bonding patterns, it's very important that the hydrogen bonds have to be more. And whenever there's hydrogen bonds and hydrophobic bonds in the form of van der Waals forces, they make a much stronger association. Ionic bonds makes it a less associated one. So it has been seen that we almost have nil ionic bonds and good amount of hydrogen bonds, and these darker hues shows that multiple places that they have binded to. It's not, see, it's the binding is in nanoseconds. It's a process where it's the entire thing happens like it's a reel of thing that is happening. So once it starts from 0 nanoseconds and go to 500 nanoseconds, they are binding, they're releasing themselves. Again, they are binding, they're releasing themselves. So, but the way they bound to different moieties, that is important. So when we see this, the certain important amino acids that are taken care of and that they are binding to that particular thing, that is what makes it important, and that also says that how this introduction of small molecules which can bind more strongly to the HC receptors, not letting the spike proteins to bind to it. So basically, when we come to the conclusion, we say ACT receptors have two main important domains. One is the functional domain and other one is the regulatory domain. SARS-CoV S1 proteins definitely bind to the functional domain, and this participates in the entry part. And if the small molecules goes and binds to this functional domain, it acts as a modulator. So these modulates the binding capacity of the spike to the ACT receptor, and we have seen that butane, the entire docking score of all the things is not more than minus 3.86 kilocalorie per mole, which is a perfect docking score. Anything beyond 5 is a good score, and it has almost 7.38, and it also follows all the 5 Lipinski rule of the ADMET, and we can also see that it makes good connection with hotspots A and hotspots C, as I have already shown it to you. That means it makes a good connection with good fluctuation points at certain important amino acids, letting not these spike proteins bind to it, which is basically binding to the hotspot B and C, and thus I can say the butane makes a conformational dynamic exchange and helps in it can be used as a drug molecule. Now, what are these? These are nutraceuticals, and these are at a very naive stage. We just talk about them. It can be taken up like now when we are back to our labs and all. We are now hoping to make these molecules and look it into how they actually going for the biological efficacies. Now, whether they have antiviral or antifungal activities or anything, that has to be checked. But before you go for any of these biological activities, the first thing that with any molecules which has been synthesized or being characterized or by being taken out from any natural sources should be first let to see whether they are actually acting as a drug-like component or not. So this is my work. It is very small, a very crisp work during this COVID time, and this is the paper that we had just published in Journal of Molecular Modeling. I am thankful to my team here. Neha is the one who had given me all these components, the 32 components, and we had worked together. And the later part of the MD simulations, we couldn't find anyone in Delhi. In fact, we got help from somebody, it's Simon, who works in, he's in Pisa University in Italy, and he relentlessly helped us in doing the last part of the MD simulation, because we wanted to actually see how much docking started and how much the binding of which are the major important amino acids are there in the MD simulation. Without the MD simulation, we wouldn't have come to the conclusion. So I thank my team, and this is the paper that we have just published. And it's, of course, it is almost negligible to the work. Dr. Bhatt and Dr. Lalita said they have done a very wonderful work, and my work is just a little bit which we could do it in the times of pandemic. And thanks for your kind listening. And we also did some work with the capabilities. I didn't show this result.
One should never be proud of itself, but to the work they have done. So always feel proud of it.
Yeah, but sir, as I said, yeah.
Whatever the work you have done, feel proud. Any question, please?
Nice presentation, ma'am. Ma'am, you have done the docking with the ACE2 receptor, and we know that spike protein is continuously changing by the mutation, so that's why number of variant of SARS-CoV-2.
See, I tell you, coronavirus as such, see all these studies, when we work on all this, the PDB files that we got, it's just not today's work. SARS-CoV was there from 2010 onwards, and people have been working on them. Many papers are there. So all the PDB files or CTX files that we have retrieved from the literature was basically based on the work that has already been done. So there are main important proteins, there are 3 important proteins, SME, out of which S1 proteins does undergo mutations, but the main important proteins that are involved in the docking are still very stable. Sorry, sir, मैं जरा उनसे बात कर रही थी. Okay, another thing. The major mutations that have taken place, if you can read through the literature, it has taken in the non-structural proteins. Okay, so these are NSP proteins, and NSP13 if you go through. NSP13 is one particular protein which has undergone much. Sarji, बच्चे को जरा बता रही थी. So that has undergone much mutations. So when we talk about the delta and the Omicron variants, that is where it is. The spike is still the same, and there are, if you go through all the literature, all those 4 hotspots has not changed. Surprisingly, it hasn't changed. So the changes are there in the other non-structural. There are 17 to 19 non-structural proteins in this thing. So whenever you're looking for a drug molecule, you have to look for those proteins which has maintained the structures. So SARS-CoV has been always there. The work has been all the computational work, all the works that has even Bhatt can say this conclusively. Most of the work we do is already based on the work that has been done on SARS-CoV. SARS-CoV-2 is the recent one, but the structure has not changed. Okay.
Was your question complete? That's what the question you were asking, or something else?
I'm just curious about that part, that spike is continuously changing, so maybe that compound which is showing in the docking result, it binds to the binding sites and inhibit the binding with the ACE2 receptor. But when the mutation is happening in this spike, then maybe that binding site due to structural changes get altered. So that's in that condition, maybe that compound is not able to bind.
That's what I said. No, the spike is not changing. That's what I said. That all the 4 hotspots is not changing. That's still there. That's what
And that molecule is binding to human ACE2 receptor.
Yeah.
So that molecule will still bind there, but your concern is also valid. Her concern is also valid because now changing the thing, mutations are happening.
Yeah, definitely.
So the drugs will definitely, sensitivity to the drugs will definitely change.
Thank you, sir.
Soma, one good work during that pandemic time. Do you have any plans to interact with people where wet?
Yes, sir. We are doing it now, sir.
Because that's why.
So yeah, we are now extracting the
modeling studies give you some idea.
Bilkul, sir.
And we hypothesize. But many things, microenvironment keeps on changing.
Bilkul. So in vitro in vivo studies are important,
I think
and now
we should start on the other side.
So
of
we
it.
have done already. We started, sir.
Any other questions? I have a question.
Yes,
Like in our body, situation changes. Like sometimes we have high fever, sometimes low body temperature, and so on. pH changes, glucose changes. Do you think these conditions can also affect the
Yes,
binding?
sir. As I said, so whenever there is a binding thing happening, it's mainly the H the hydrogen bonds is important, and there are small ionic bonding, which are also known as non-atomic bondings or ionic bondings. Now, ionic bondings do affect through the pH and the temperature. As here you can see the bonding is only at one position. But there can be certain other changes because these are very flexible. These are proteins and they are very flexible. So they definitely undergo changes during high fever or the change in the pH.
But that can affect the efficacy of this drug.
No, sir. We have just seen the docking strategy and now we have started extracting it. And we are doing it in green synthesized, like that it's being synthesized in nanoparticles and the green form. So we are now looking into it.
And if there is no question, I think we should slap some
As ma'am said that she checked with the looking screw. So ma'am said that you checked with the looking screw. Then that is the addition to the answer to of the ma'am. If it's already checked with the looking screw, that means it's more favored to be the drug. And drug to be more favored as maybe the pH change or the other factors. It will be favored is as a drug.
Actually, sir, is right. Admit, अगर आप करते हैं ना, so actually they are not 5. We go by 5 lipid scales. They are about, say, I think lipid scale में कितने हैं sir? 5 तो 5 तो हम देखते हैं, but when we do that, there are more than 10 other characteristics. So उसमें से हम 5 choose करते हैं. तो जो best होते हैं, जिसमें और बहुत सारे changes जैसे lipophilicity, of course the change होगी आपकी pH से. और आपकी हाइड्रोफोबिसिटी वो भी आपकी चेंज होगी तो वो तो होगी पर जहाँ पर हम देखते हैं कि ड्रग्स 5 जो दे आर एक्चुअली एग्रीइंग विद दोस थिंग्स वी गो फॉर दैट
Just a small addition, there is called as druggability.
Yeah, druggability quotient.
The druggability. That's how the
Yeah, it's known as druggability quotient. Yeah.
Thank you very much.
Yes.
Insightful lecture.
हम लोगों ने नाम चेंज कर दिया। आप आइए।
Sir, this side. Sir, there is a banner.
Banner
बैनर जरूर चाहिए।
अच्छा banner.
All the best.